Hello, good afternoon, everyone. Let's get started. Today we will be taking up the study guide for lecture 1, which is scientific method. And I posted a link in the chat box for those of you who have not had a chance to download the study guide. It will take you right to eCentennial, where you will have to log in and you can get a copy of it. So ideally, you can print this out and do it along with me. And if you don't have a printer, you can just write it down the solutions in your notebook as we do it. You do not have to hand it in to me. This is for you to study for the purpose of studying only. Okay, it's not for any marks. And remember, we only have live class on Thursdays, not Tuesdays. Okay, Tuesdays is reserved for you to do the asynchronous learning. Speaking of which, you should have already watched the recordings for lecture 1. And if you haven't already, then some of the stuff I say might not make too much sense because the tutorial, right? It's not meant to be a substitution of the recording. It's more like a recap. Okay, so you really still have to watch the recording to get the full picture. Okay, this is kind of like, you know, those things on YouTube's where they shrink the whole movie into like 15 minutes or something. You got to watch the whole thing to, you know, get a feel for what the lecture is about. In any case, we will begin right now. This is of course recorded, and so if you need to watch it again later, you will have the option of doing that as well. And then the notes that I'm filling in right now, I will also post a copy on eCentennial afterwards. So, you know, if you missed anything, you can go back and get it. So here we go. List 7 characteristics of living organisms. Okay, so living things, they're very diverse, they're very different, but at the core of it, there are some features that are common for all of them. So the first one is they exhibit order. Okay, exhibit order. So order means there are some sort of complexity to it, right? If you think about like the human body, right? Very complex, many moving parts. And there is some kind of symmetry usually, right? So all these things is evident in all living organisms. Okay. Another characteristics is the ability to maintain homeostasis. Okay. Can someone explain to me what is the meaning of homeostasis?
It means everything is like balanced, even.
Yeah, very good. Okay, so this homeostasis means an internal balance, internal equilibrium. Okay, which basically means balance. So an example of that would be our ability to maintain a body temperature of 37 degrees Celsius. Okay, but there are so many other equilibria in the body. Okay, your blood sugar level, your blood calcium level, right? All those are examples of homeostasis. And all living organisms, regardless if they're complex like a human being or simple like a single cell organism, they have to be able to maintain homeostasis. Now, there are 2 types of mechanisms or methods that is used to maintain homeostasis. Okay, there are those that are physiological, okay, and then there are those that are behavioral. Okay, so the physiological example would be it's very cold and then you shiver. Okay, as you're shivering, your muscle contracts and it brings your body temperature up. Okay, so that's something that your body does for you automatically when you're cold. Behavioral mechanism would be like putting on a jacket when it's cold. Okay, so one, it's sort of like deliberate action, and the other, it's kind of automatic. And other characteristic of living things. These are not in any particular order. Okay, I'm just writing it as I remember them. Okay, so growth and development. Okay, so the organism is able to grow, to get bigger, perhaps, right? And develop. Okay. Sometimes, if you think of like a butterfly, the caterpillar looks very different than the actual butterfly, right? Different stages of development. Same thing as human, right? Think about like a baby and how grown up looks like. There are lots of development going on. Some are external features, some are like internal, right? Your brain form new connections. All those things are example of growth and development. Another one is they require energy. Okay, so we are consumers. That means we have to eat in order to obtain energy. Things like plants, they are producers. So they would harness the energy from sunlight and make their own food. But regardless of how you acquire your food source, okay, there is some kind of requirement for energy for living organisms. Another one is it will respond to stimulus. Okay, so stimulus is basically like changes in the environment. Changes in the environment. Okay, so you know, if you take a stick and then you poke at a bug or something, then the bug would move away. Sometimes they curl up, right, into a ball, those little pill bugs, right? That's an example of response to stimulus, right? They're able to have a particular response when certain changes takes place in the environment. What's another one? Anybody want to give me another one? You can either type in the chat box or you can unmute.
Reproduction.
That's pretty good, right? The ability to reproduce. That's kind of like the goal of all living organisms, right? To reproduce, to make more copies of themselves, right? More offsprings. Okay, so that one is pretty self-explanatory. And the last one is the ability. Sorry, go ahead.
Is it cells possibly? No.
That they are made up of cells. Yeah, they are made up of cells. Very good. But I don't think that's one of the characteristics I included, although you are absolutely correct. The last one is the ability to adapt, right? Robinson is saying that in the chat room. Very good. So adapt and evolve. Okay. So I just want to clarify what evolution means. Okay, it's important to know, to remember, right? I'll put a star right here. Population evolves. Population evolves, not individual. Okay, population evolves. So what does that mean, right? Let's have a hypothetical situation here. Okay, let's say there is an island, and on this island there is a type of organism that looks like this. Okay, so these little things with two legs, they come in two forms. Okay, some are naked, so to speak. There's no fur on them, and then some of them they have fur. Okay, so these are the furry ones. Okay, and then they are living happily on this island. And then what happens is, let's say there is some kind of environmental crisis, and it causes the temperature to plummet to say -60 degrees. Okay, and it's a constant thing. Okay, it's going on for like years and years. So what's going to happen is, well, the naked ones, the ones that does not have the fur, they are going to be cold and they might not survive as well as the furry ones. So they will die off. Okay, not all of them. Okay, maybe a few of them will, remain.
Is that survival of the fittest?
Exactly, right? Yeah, this is precisely what it is. So the furry ones, they are going to be able to survive better in this environment, and then they will be able to have more offspring. And so over time, you will end up having more of the furry ones, okay? Because those are the ones that are surviving and having more babies, right? Okay, so if you compare the population, the initial population, with the population that you get like after a year or 2 years, you will find that more individuals in the population would have the fur, right? Compared to what you started with, right? Because they possess the trait that would allow them to survive better in this particular environment, so that the entire population has evolved, okay? Individuals do not evolve. So it's not like this. This is a big misconception when people think about evolution. They think that there is the naked one, and then it gets really cold, and so they start growing fur. Okay, so that is not what happened. Okay? Individuals do not evolve. It is the population that evolves. Okay, and in order for this to happen, that favorable trait has to be there within the population to begin with. Okay, it's not just going to come up out of nowhere.
Sir, I just have a quick question.
Mm-hmm.
So I was reading something. Basically, it said that people who like aren't vaccinated, don't have like immune antibodies, like down the line that they would eventually like die out. Is that true? Like down the line, not further, they may not die. Let's say to people who have no type of vaccinations, like polio, measles, or whatever it may be, down the line, would their generation die out? Or
I that is an interesting question, and it's not something I can answer in a minute or two. But no, that is good. That's very interesting, actually. They have a higher chance of not surviving, right? Let's say someone is completely, let me give you an example. Have you guys read about this? I think I read it about like a year or a year and a half ago, that there is this remote island, right? That is a very remote place that no one has ever gone to.
yeah,
okay?
and they don't let people come on the island either.
That's right, and that's what their fear, right? Because those people are so isolated in that island that they have no exposure to any of the modern diseases, and so if someone goes there, right, and try to engage with these people, they would spread the what we have to them, right, and potentially wipe off the entire population on that island, right?
Yeah.
So that's kind of similar to what you're talking about.
Okay.
But what you're saying is more on like a global level, right? If there's a bunch
Yeah.
of people who do not get vaccinated, will they all die out? They might, but it's not evolution, right? Because look, if I am vaccinated and my kids are not going to automatically be born with all the stuff I'm immune to, right? You know what I mean? Like
Yeah.
that immunity is not permanently transferred, right? So it's not exactly related to this current situation. Nevertheless, excellent
Thank
question.
you.
Moving on, we have here what we call the induction and deduction, which is basically like two different reasoning approach that we can use in science. So for induction, what that does is we derive, okay, we come up with, right? We derive a general statement based on many specific observations, right? Or examples. Okay. So that is going from specific to general. Okay. So to draw it in a diagram, it's like this. Okay, I make one observations, and then I make another related observations, another one, and another one, and then many, many, many more. And then you take all these observations that you get, and then you're gonna form some kind of general statement. Okay. Sorry, guys, just give me a second. My daughter is she's doing online learning. I think she needs some help. I'll be right back. Okay. And I'm back. Okay, so that's what induction means, going from many specifics to general. So an example of that would be like, let's say you observe that birds are made up of cells. Okay, and you're like, cats are made up of cells. Okay, and then like, I don't know, sharks are made up of cells, right? Plants are made up of cells. Okay, so you make all these observations. Okay, they're all made up of cells, and so you come up with a general concluding statement that all living things are made up of cells. Okay, that is an example of induction. Any question? Okay. On the other hand, deduction or deductive reasoning start with some kind of general statement, some kind of general premise, and you're going to use that to apply it to specific cases to sort of make a prediction sometimes. Okay, so this goes from general to specific. Okay, using the general statement to make predictions. Okay. An example of that would be starting with, let's say, this statement, right? All living things are made up of cells. Okay. And then you travel to that hypothetical island that I was talking about earlier, and you discover these circle things with legs, and they're like, oh, they are living things. And since all living things are made out of cells, then these little thingies must also made up of cells. Okay, so again, you start with that general statement. And then you're going to apply it to different situations. Okay, so on and so forth. Is this okay? Any questions? Okay. Well, let's do a poll question and see if you will be able to convince me that you do understand. Go ahead.
Sir, would A be
You
false?
can use the poll to answer. Do you see the poll that I just?
Yeah, would it
Yeah.
it would just be like A or
Well, let's you can choose your pick and then we'll talk about it after, so other people can try as well. Thank you. Okay, has almost been 2 minutes. Just gonna give it a few more seconds for people who wanna try. If you're not sure, you can choose E, so that way I know that you're there. Just not sure what to choose. Okay. So let me share the results. You can see that we have a lot of people choosing A and C, right? And that's typical. That's what most people choose when I give them this question, either A or C. So let's talk about this, right? Deductive reasoning. Okay. So what is deduction again? Deduction is you start with a general statement, right? General statement, and then you go to specific, right? Okay. You start with general to specific. Okay. So let's take a look at what these things are saying. First one, all virus requires a host to replicate. That sounds like a general statement, right? So that could be the answer. Okay, this is a general statement. Okay, so maybe I'm just gonna keep reading first. Next one, COVID-19, Ebola, influenza, or viruses that requires a host to replicate. So these are like very specific examples, right? And then you're going from specific to general, which is inductive reasoning, not what we are looking for. Next one, all virus requires a host to replicate. That again is a general statement, so possibly the answer I'm looking for. Next one, COVID-19 is a virus and it requires a host to replicate. Ebola is also a virus, therefore it also requires a host to replicate. So these are just like two separate observations, right? Two separate examples. We are not generalizing anything from it, or we're not applying to many specific cases. So that is also not what we are looking for. So far, so good, I hope. Now, which is it? Is it A or is it C? Well, let's take a look at the first one again. All virus requires a host to replicate. COVID-19 is a virus. It must require a host to replicate. Whereas this one, it says COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. So at first glance, they are very similar, okay? But you really have to think about the logic, like how the logic is flowing in these statements. Let's take a look at C again, right? It might be a little bit hard to follow when we talk about viruses, COVID and replication. Let's try to use this same format on something that we are more familiar with, okay? So instead of saying virus, I'm gonna say all birds. Instead of virus, I'm gonna say birds. All birds have wings, okay? So have wings is like require a host to replicate, okay? And then COVID-19, I'm gonna say bees, okay? COVID-19 requires a host to replicate. So going in the same format, I'm gonna say bees have wings, okay? So therefore what? What I'm gonna write next in this format? What's my conclusion?
They can fly.
But
Birds has
okay,
wings.
yes. Say that again, second student, please.
Birds has
Um,
wings.
Birds
They
have wings.
Yeah.
Who has wings? What
Penguins
has wings?
can't fly.
Birds.
Okay, so here, right? Yeah, okay, thank you. Sebastian actually have what I was looking for in the chat room, right? First of all, right, this sentence mentioned nothing about flying, so we're not gonna put that in. And secondly, if you follow the logic, you see all birds have wings, bees have wings, therefore bees are bird, right? Do you see that? That's what this statement is essentially saying, right? They're saying, okay, all viruses requires host to replicate, and then COVID requires host to replicate, therefore it must be a virus. That's what the logic sounds like if you use something like birds and bees. That is why this is not the correct answer. Okay, so this is the correct answer. Okay, so it would be like, all birds have wings, and then something like a sparrow is a bird, it must have wings. Is that okay? Anybody have a question?
I like your other explanation better, if I'm gonna be honest.
What's the other explanation?
I think you said it starts off as like a general idea and then it goes into more specific understandings.
No, I understand that, right? But you see that the dilemma here that students are having is that these are both from general to specific, right? Do you understand that?
I see the C as a repetitive because if you look at the first one, the first two are the same. Like, yeah, all viruses require a host to replicate.
Mhm.
Okay. The second one, the second C, it just repeats it. COVID-19 requires a host to replicate, therefore it's a virus. But A explains like, okay, all viruses need a host, number one. Number two, COVID is a virus. And number three, it requires like, just the way, I don't know if it makes sense.
Okay, so I don't I don't want the people to be missing the point, right? Both, and I understand what you're saying. Both A and C are going from general to specific. However, what I'm saying is the logic in the third one is not correct, right? And you can see why it is not correct using the bee and the birds example, right? Okay, because the ending, look at the ending, right? It's different. It says COVID is a virus, but here it says COVID requires a host to replicate. The general statement states that the virus requires a host to replicate. Okay, not the other way around, right? Not like everything that requires a host to replicate is a virus, right? You see that the order of the wording actually matters in this case.
Got it. Thank you. I would have
Okay.
failed the test then.
No. So just a little word of comfort. Not every question is going to be as intense as this one. Okay. Remember, there are like normal multiple choice questions that are factual based, right? Just recalling information. And those are going to be much easier. Okay. But that's what these tutorials are for, right? To help you prepare. Okay. I wouldn't be surprised if you see this, something like this on the test, like similar to this. Okay. So make sure you have a good understanding of it. Okay. All right. What was I talking about? Yes, let's go back to this. We have the scientific process. Okay. So the scientific process have many steps in it. And we are going to list them out and, talk more about it. Let's choose purple. First is observation. Observation. Okay, so you're gonna be seeing things, observing things, and then that might cause you to ask some questions. Why is it the way it is? Okay, questions, observations, and from those questions, you're gonna try to come up with possible explanation, right? To explain why it is that way or what is it that you're looking for. And these tentative answers, which may or may not be correct, they are called hypothesis. Okay, so hypothesis is basically a tentative answer to your questions. Okay, and in order for the hypothesis to be valid, number one, it must be testable. Number two, it must potentially be wrong, and we call that falsifiable. Okay, potentially wrong. Okay. You learned that hypothesis is the first. Yeah, I mean, it does usually, well, not usually. You cannot have a tentative answer to something if you don't have the question, right? Okay. And observation can also occur during the experimental phase. Okay, but here we're talking about just random observations that you make. Okay, so you observe that frontline healthcare worker gets sick more, right, due to COVID. That's an observation. Okay, you're not doing any experiments per se, right? And the question you ask me be like, oh, why? Why is it happening? And then you theorize that, oh, maybe it's because they don't have enough PPE, right? That's a tentative answer. Okay, then you're gonna make a prediction. Okay, you're gonna make a prediction. Okay, if my hypothesis is correct, then it should be blah blah blah. If it's not correct, then it should be blah, blah, blah. And then from that, you will do an experiment. Okay? And then to see if your experiment agrees with your hypothesis or not, then you can conclude. Okay, conclusion. Okay, I think in the recording, I gave you an example of a flashlight, right? You pick up a flashlight, turn it on, you observe that it is not working. And the question is, why is it not working? Okay, your hypothesis would be, maybe it's out of battery. Okay, so the prediction is, if I replace the battery, it should turn on. So you go ahead, replace the battery, and then that's your experiment. And then you turn it on, it works. So your conclusion is, the problem was you didn't have enough battery. Okay, so that's an example of how the scientific process work in a very simple sense. Okay, so another thing that people make mistakes is the hypothesis is a tentative answer. So it is not a question, not a question. Don't write the hypothesis as a question. So in the lab that you will do later on, I'm going to ask you to write some hypothesis. Just make sure you are writing a statement rather than a question. You don't answer a question with a question, right? Okay. So as I mentioned, in order for the hypothesis to be valid, it must be testable
sorry.
and yeah.
Sorry, is hypothesis is similar to statement?
Say that again. I'm sorry.
Is a hypothesis is similar to statement?
Hypothesis is similar to what statement? I'm sorry.
Statement.
Could you
I guess
please
she's
type?
asking, is a hypothesis equivalent to a statement? Like, is it a
Oh,
statement?
yeah, it is a statement. Yes, it is a statement. Thank you for clarifying. Yeah, sorry. Yes, it is a statement, right? Not a question. Okay. So.
Are you allowed to also say a hypothesis is an educated guess, or?
Yeah, I know. People are like, especially like in high school, they like to say that. I personally don't really like that term because it's not really. It's not guess to me. It's more like, you try to formulate an answer. But sure, educated guess, why not? Okay, so for example, right? If I want to know whether vaccines will help protect you from COVID, okay? Okay, so the hypothesis would be vaccines reduce the risk of catching COVID-19. That's a hypothesis, okay? I will not say, will vaccine reduce the risk of catching COVID, okay? Because that's a question, right? Okay, you want to write it as a statement, not a question. That's all I mean. Okay, I hope that clarifies it for you, Sita. Okay. Give an example of a non-testable hypothesis. Anyone wants to try? Feel free to type it in the chat box. Or just unmute. Give me an example of a non-testable hypothesis. Okay, so we have things like angels do not exist, cultural beliefs. Yeah. Okay, so yeah, so those are good examples, I suppose. So cultural beliefs like or like any kind of beliefs, right? Personal beliefs, right? Any kind of opinion, right? So things like biology is harder than math. Okay. That might be true for you, but not necessarily for another person. And there's no way to test it. Okay, it's a personal preference, right? Okay, and so there you have it. Beyonce is the best singer. Yeah, sure, I like that one. I'm gonna write it down. Okay, Beyonce is the best. Okay, Kanye West might agree, but other people might not. Okay, I don't know, right? Okay, what's another example? Give an example of a non-falsifiable hypothesis. What is that? Anyone? Okay, non-falsifiable. Remember, for the hypothesis to be correct, it must be potentially wrong. Right? So if you have a statement that cannot be shown to be wrong, then it's not a good hypothesis. Okay, and by the way, right, I just want to clarify, I'm not saying any of these statements are true or not true. I'm just saying that they're not good hypotheses, right? Anyone? Non-falsifiable hypothesis? Yeah. Sierra said God created the universe. Yeah, that one is both non-testable and non-falsifiable. Alien exists. Yeah, could be. The sky is blue is it's kind of a fact though, right? So not sure if that one would qualify as non-falsifiable. Here is my favorite one. Okay, I hear this from my parents a lot when I was growing up. Okay, so they like to say this to me. Okay, son, if you work hard enough, then you will get A+ in whatever. Okay, so
That is a
growing up.
lie.
That is a lie. Growing up, the problem I had was English. Okay, so they would be like, if you work hard enough, then you will get A+ in English. Now this is a non-falsifiable statement, non-falsifiable hypothesis. Okay, because if I work hard and then I don't get A+, and then I come back and say, hey, what happened? Then the person could always counter argue and say, well, that's because you didn't work hard enough. Okay, so I can work harder, and then if I still don't get A+, then they could still say, well, that's because you didn't work hard enough. Okay, so something like this is non-falsifiable. Okay, there's no way for it to be proven incorrect, and therefore is a bad hypothesis. Okay, hope that makes sense. We're going to move on now to the next part. Let me just remind myself what the next. Here, let's do some easier poll question to help you guys feel a little bit better after that very intense first one. Okay, go ahead. Okay, I will end the poll. Yeah, most people got it, right? That's what we've been talking about. Tentative answer is a hypothesis. Okay, what about this one? What about this one?
Can you see what we answer, or is it all anonymous?
It's anonymous. Okay, so don't worry. Okay, you can answer it, and if you get it right, get it wrong, I won't know who you are. In fact, I don't even know who you guys are just by looking at the names. There's no faces, right? You know what's gonna be weird? If we return to campus in the next semester, you would totally recognize me in the hallway, but I would have no idea who you are. That's a little bit creepy. Okay, the answer, again, most of you got it, which is very good. It's experiment, okay? So that's what you will conduct to test a hypothesis, all right? Okay, so like I said, right, the quiz would have some of these more straightforward questions. Not everything is going to be very involved. Okay, moving on. We have give 2 examples of okay, there's a question, there's a private message. Why is data not included in the scientific method? I think it's embedded within the experimental the experiment portion. When you conduct an experiment, you're going to make observation, you're going to collect data, you're going to analyze the data, right? All that kind of falls within the experimental part. So we're not listing it out separately. Okay, give 2 examples of primary sources and secondary sources. So primary sources is basically first-hand recollection of an event or first-hand description of something that you've done, right? So an example of that would be like a peer review article, okay, journal. And so when researchers do research, okay, they do experiments, they collected the data, and they write an article about it, right? That article is then scrutinized by other experts in the field before it gets published, right? So that is a first-hand article about what the experiment is about. So that is primary source. Yeah, interview, that is correct. Okay. If there is a reporter doing live reporting, like a journalist doing live reporting at a scene of a car accident, perhaps, right? That is primary source. Documentaries, you know, sometimes it's a little bit difficult to distinguish primary or secondary. If it's a documentary based on you know, footage that you've obtained by yourself, right? It could be, but if it's a documentary that includes multiple sources, then for those portions of the documentary, it would be like a secondary source. Sometimes it's not very clear cut, right? So as for secondary sources, it would be things like textbooks, right? Blogs, newspapers, okay? Things that are just recounting events based on someone else's, okay, experience. Books, right? Your biology textbook, right? If the textbook tells you living things are made of a cell, the author of the textbook did not came up with that, right? Okay, he learned it somewhere, he read it somewhere. So that's a secondary source. Okay, and again, articles, where someone asks, is article primary or secondary? It depends on the nature of the article, right? Like I said, if I did the experiment and then I published the result in an article, that is a primary source. If my article is about someone else's work, then it could be secondary sources, right?
I have a question. With technology evolving nowadays, what happens with like digital newspapers and stuff like that? Are they considered like air quote blogs or articles still? Are they considered like primary sources?
Again, right, like it's not so much that it's digital. I think like I could write an article myself, come up with an article myself completely, and then post it online, right, onto the blog, right? That could be a primary source. Okay, but if I'm just putting a blog and like reposting someone's work, right, it's or like, you know, putting someone else's like a link to someone else or whatever, someone else's article, then that could be a secondary source. Okay,
Okay,
so thank you. I think the point here is it's not very clear cut. Okay, it's not so much the actual material, but the thing you have to ask yourself is whether or not the person is the original person who came up with these concepts, right? The originality of it is what distinguishes it from a primary source from a secondary source. How about gossip? I don't know. That one is so tricky. I haven't really put much thought into it. Maybe you guys can figure that out. Okay, next part. In each of the following experiments, state the independent variable and the dependent variable. Okay, so this part is done in the lecture recording. I'm not going to go through it again. I will, however, explain what independent and dependent variable means again. Okay, so independent variable. When you do an experiment, the independent variable is the variable that is changed or manipulated by the researcher on purpose. Okay, so first of all, a variable is something that can be changed. Opposite of a variable is a constant. Okay, so there are some things you want to change in experiments, and then there are some constants that you want to keep unchanged. Okay, so the variable that you deliberately change in an experiment, that is what we call the independent variable. Okay. The dependent variable, on the other hand, that is the variable being observed or measured. Okay. The point of an experiment is to determine whether or not changes in the independent variable will have any effect on the dependent variable. Okay, so by changing the independent variable, you want to see if the outcome, the dependent variable, will be affected. Okay, so if I'm testing the effectiveness of a vaccine, okay, I would give some people vaccine, some people no vaccine, and then I would try to see if they get sick. Okay, so by changing the vaccine status, I want to assess whether or not it will have any effect on the dependent variable, being whether they catch the illness or not. Any question about that? Okay, so I have someone who messaged me and say more examples. Sure. Okay, let's use the example in the lecture. Okay, so let's say I have plan. Okay, plan number 1 and plan number 2. Okay, and then I want to see if sunlight will make them grow taller. Okay, so I put one plant in the sun, and then one plant I will cover it with some kind of shading. Okay, what am I deliberately changing? I am deliberately changing whether or not the plant is exposed to sun. Correct? Okay, so in this case, the independent variable is exposure to sun. Okay, in other words, yes sun or no sun. Is that part clear? Okay, what am I measuring? What am I trying to observe? I'm trying to see if one plant will grow taller compared to the other, right? So the dependent variable, the variable that I'm trying to measure, right, either with a tape to see how long it grows or to observe, just visually see which one is taller, that would be the dependent variable, okay? Height of the plant. Okay, so the whole premise is this, the whole idea of doing an experiment is this. You're trying to find out whether or not, you know what, let's just make it even simpler. Will changing the independent variable have any effect on the dependent variable? Okay, so by changing the amount of exposure to sun, will it have any effect on the height of the plant? Okay, I will give you another example. Let's say I want to find out whether or not doing these live Zoom meeting is beneficial to students. Okay, so what I could do is I could have two classes. Okay, one your class and my other class. For you guys, I will do this virtual tutorial every week. For the other class, nothing. Okay, they just do everything online themselves. Okay, so that would be independent variable. Having virtual meetings or not. Okay, what would the dependent variable be in that case? Well, I can see what is the final class average afterwards, right? If I see that the people in this class do better compared to the other class, then I can conclude, oh, having the live tutorial actually helps them do better. Okay, so the dependent variable in this case would be the final grades of student. Is that okay? Any follow-up questions before we move on? All right. Okay, so here we are going to talk about the experiment by Francesco Redi and Louis Pasteur. Okay, so first, there is the spontaneous generation hypothesis. Okay, so to keep it very simple, spontaneous generation basically states that living things came from non-living things. Okay, so people believed that in the past that living organisms would come from non-living things. Okay, and an observation that supports that is when a piece of meat is left outside, flies and maggots will appear on the meat. Okay, so if I didn't know any better, and then I see that, and I leave a piece of meat outside, and then I come back 2 weeks later, I see like maggots and fly buzzing around, and I'm like, oh, maggots and flies, they must have spawned from the meat. Okay, so that is the essence of spontaneous generation. Okay, there are other weird phenomena too, like fish raining from the sky. It's actually more common than you know. I actually saw it on my news feed just a couple of weeks ago. I forgot where though. That it happened. Okay, fish were raining from the sky. So people look at that and then they're like, oh, spontaneous generation. The living thing, the fish, came from the sky, which is a non-living thing. Okay. So he's gonna do an experiment to test this hypothesis. All right. So he has 3 groups. One group, there's going to be a jar, and it is going to be an open jar. And then the second one is going to be a jar with a netting on top. Okay. So again, I hope you're drawing this with me. No sense watching me do it. You must also do it. Right? The act of copying things down actually helps you learn, believe it or not. And then the last one, he's gonna seal this jar. Okay, he's gonna seal it. There is a sealed jar, and then here is a jar with netting. Okay, and then he's gonna put a piece of meat in it. Hopefully he had the sense of putting the meat in before he put on the netting or the jar. Okay, so there's going to be the meat. Okay, and then he's going to leave this thing outside for, I don't know, like a week or something. I'm not sure how long. So in this experiment, what would the independent variable be? Someone please help me out with that. What's the independent variable? What is the one thing that he deliberately changed in the experiment?
Group 2, independent.
The independent variable. Yeah, which one is it?
Group 2.
I'm sorry. Could you one more time?
Is it group 2? Jar with netting.
The type of jar being used, right? Exactly. Yeah. So that is the independent variable. Type of jar, right? Are we using an open jar, a jar with netting, and a sealed jar? Okay. And then the dependent variable. What is the dependent variable? Anyone? What is he trying to measure? What is he trying to observe from this experiment?
The meat?
And what is he trying to find out about the
Flies.
meat? Sorry?
Flies.
The
The
flies and
flies.
maggots.
That's right. He's trying to see the presence of flies and maggots. Okay, so he deliberately changed the type of jars, and then he's going to observe to see what's going to happen. So what were the results of his experiment? Okay, as you might expect, okay, either you use common sense or you watch the recording. After some time, there will be flies buzzing around, okay, in the first one, and there will be like maggots crawling around. Okay, maggots. There's the maggots. Okay, so jar number 1, maggots plus flies on the meat. Okay, this must be a pretty gross experiment. I personally is very afraid of maggots. Like every summer, there is like at least one incident where I try to take out the garbage and then it's full of maggots, and it's really, really gross. My wife's not bothered by it though. So I just call her and then she would come and clean it up. She'd be like, I don't know what's so gross about the maggots. They can't fly, they just crawl around. Anyways, number two, we would have the flies. Okay. And then the maggots would be on the netting, but not the meat itself. Okay. So maggots plus flies on the netting, on the net. And number 3, nothing, no flies, no meat. Okay, probably can't eat it though, but at least there's no flies, no meat. Okay, so that was the observation. What would the conclusion be? Well, as you can see from this experiment, that the meat does not spawn flies or maggots, because if that's the case, you would also be able to see the maggots and the flies inside the sealed jar, right? And that did not happen. So the conclusion is that meat does not produce flies and maggots. Right? Only flies produce maggots and flies. Okay. I mean, you look at something like this and you're like, obviously that's the case. You didn't have to do an experiment to find out. But remember, this was a long time ago, right? So you know, we didn't really know a lot of things. Does hypothesis? So yeah, so you know, his hypothesis is that when a piece of what observation led to us. I didn't write a hypothesis here, but then based on this result, he rejected the hypothesis of spontaneous generation. Okay, so therefore, the theory of spontaneous generation is rejected. Okay. So then there is another guy. His name was Louis Pasteur. He's the same person who came up with the pasteurization process. Do you guys know pasteurization? What kind of food in your fridge should be pasteurized?
Milk.
That's right,
But
milk
there's a
and
whole
dairy.
debate saying you can drink unpasteurized milk. Is that true?
I think you could, but I wouldn't. It's like you might get sick from it, right?
My dad
Since-
loves to brag about, like, you know, when he was on the farm. Oh yeah, I can just drink milk straight from the
Straight
cow.
from the cow, right?
And I'm like, what the heck are you talking
Yeah,
about?
it's yeah. Some people do that, and it's fine. Some people have stronger immunity than others, right? Same thing as honey, right? Some honeys are, you know,
The way you
unpasteurized,
say
and some
honey
are
reminds me
pasteurized.
of the video.
What's that?
Too much honey.
Okay, I'm not sure what that was about.
I wouldn't
but-
know. I can't eat honey.
Yeah, so some honey is pasteurized, some are not, right? And again, you should give the pasteurized ones, especially to kids. Okay, so he's the same one who came up with that process. In any case, how is he related to what we're talking about? Well, at the time, some people, okay, let me choose a different color so it looks different. So some people didn't believe Brady's experiment. Okay, some people still think that spontaneous generation occurs on a microorganism level. Okay, so they're like, okay, sure. The flies and the maggots, they're like big, so maybe it's not true for them. But on a microbial level, spontaneous generation could still be true. Okay. So what did Pasteur hypothesize? He says that if cells arise from non-living things, then they will appear from sterile broth. Okay, so sterile means like heat treated. Okay, everything is killed. Everything is killed off. Okay. And then he did an experiment, right? Yeah, and all this is explained, of course, in the lecture recordings. In Pasteur experiment, he had two flasks. He has a flask with an open top and then one with a swan neck. Which of these would be the control group? Let's do a quick poll to see if you guys are able to tell me. Okay, which one is the control group? If you think this is control, please choose A. If you think this is control, please choose B. Excuse me, if you're not sure, you can choose C. Which is the control group? We are neck to neck right now between this one and this one. We give it a couple more seconds for people to decide. Okay, well, looks like it is almost half-half. So let's talk about what a control group is. The control group is the group that has no treatment, no modification. No treatment. Okay, in other words, the default. Okay, that's the control. The experimental group is the one that has the modification with modification or treatment. Something has been done to it. Okay, so in this case, the flask with no special neck, that is the control. Okay, and this is the experimental group. Okay. What was the independent variable in Pasteur's experiment? What was the one thing that he deliberately changed? It is the type of opening, right? For the flask. Okay. Do you have an open top flask or do you have a swan neck? Swan neck. Okay. Next question, how did Pasteur ensure the initial condition of all the flasks were identical? Well, he sterilized the broth, right, by heating it. So heating the broth to kill off everything. So later on, if you look at it and you see some microorganism, it was not from before. Okay, so he killed off everything, and he probably used the same type of broth, right? Same conditions, as in like same temperature. You leave them in the same temperature in the room, so on and so forth. Okay. And what was the result? Well, if you remember from the lecture, microorganisms were only observed in the control flask, but not the swan neck flask. Okay. Now that should make sense, right? Because if it's an open flask, right, things from the air would fall into the broth, and then they would multiply and have lots of microorganisms. Whereas if you have the swan neck, right, all the airborne stuff will be trapped here. They won't be able to go in, right? So the broth remain sterile in the experimental flask. Okay, so for that reason, he was able to conclude that the theory of spontaneous generation is not true. Okay, because the sterile broth did not produce any cells. Okay, before we take a break, does anybody have any question about anything we've talked about so far? All right, we will take a 5-minute break. Let's resume at let's say 1:45. We'll come back at 1:45. Okay, see you in a bit. All right, let's keep going. Let's start with another poll question. Go ahead, please try this.
Your face does not look impressed at all, sir.
My face does not look what?
Impressed at all? Are the polls that bad?
No, I'm just tired. It's like I have 2 kids at home doing learning, and has been difficult. Okay, maybe half more minute. Okay. These are the results. Let's take a look. The independent, we are looking for the statement that is false, right? That is incorrect. So first one, the independent variable in this experiment is the presence or absence of maggot and flies on meat. Remember, independent variable is the variable that is being changed deliberately by the person, right? So that would be the type of covering that is being used. So as a result, this is the incorrect answer. Okay, this is the false statement. Okay? All the other statements, they are true. Okay. A lot of people choose B as well. Maybe it's just that you guys misunderstood the question. Could it be? We are looking for the false statement rather than the true statement. Okay.
Sorry, I'm misunderstood with dependent variable and independent variable. That's
Okay. Yeah. So one more time, right? Independent is the one that we have control over, the one that we can change. And then the dependent one is the one that we will be observing. Okay. One way to remember it is this. Okay. Independent. Independent is the one that you are in control of. Okay. Independent is the one that you are in control of. Okay? It's the one that you can change. Okay. Hope that helps. Hey, here is another one. Here's another one. Let's try that one more time. Please go ahead. Right, just a couple more seconds. You spend about like no more than 2 minutes on each question on the quiz. That way you can make sure you have adequate time. Okay, very good. Most people got it, right? The independent variable is going to be the type of flask being used, with the open top or the swan neck. All right. This one is about hypothesis. Sure, let's do that as well. Oh, this one is an example of a multi-select question. There are more than one possible answer. So please choose all the correct ones. Okay, pretty good. So there we go. Yeah. The first one is invalid because eating broccoli is good for you. Good is such a vague term. It's hard to test whether something is good or bad, right? Like in what sense? I need to have more details, right? So like that's not valid. Animal testing is bad. That is an opinion. This one, it's non-falsifiable. So A, D, and E, those are the bad hypotheses. Okay, we will come back to this one in a bit. Let's go back to our slide here. Okay, Long answer question. You are the lead scientist in the study to determine if taking multivitamin supplement reduces the risk of catching a cold. Let me copy this question and we will do it. We'll do it at the very end right here. Okay, because I'm going to post this online for you later in case you miss anything. So let's just put it right here. Okay, so A, write a hypothesis for the experiment, right? Does anybody want to tell me a hypothesis that they would put for this? Anybody? Okay, I will write an hypothesis. It's quite straightforward. You can say taking multivitamin supplement will reduce the risk of a cold infection. Okay, so you can either write that or you can say taking multivitamin supplement will not reduce the risk of catching a cold. Okay, doesn't matter whether you think it's true or not true. You pick a stand and you write a statement. That's your hypothesis. Okay. B, what is the independent variable and the dependent variable for your experiment? Okay, so people were having trouble with this concept, right? So let's ask the class again. What would be the independent variable be in this particular experiment? One would take the vitamin, one won't. Exactly, right? So the independent variable is taking the supplement or not. Okay, that's the one that you are going to be in control of as the researcher. And then the dependent variable, the one that you're going to be observing or measuring is the number of cold infections. Okay. Part C, what group should be involved in your experiments? So we're gonna have the control group. Control group. What are we gonna give to the control group? Nothing. Not gonna give them supplements. We're going to have the treated group or the experimental group. That's the group that will be receiving supplements. Taking the multivitamin supplements. And then because we're dealing with humans and humans are complicated, that's why we should have a third group. What is the name of that third group that we need? Anybody? That's right. We are going to need a placebo group. Very good. And what are we going to give to the placebo people? What will we be giving to? We're going to give them money. We probably will give money to all of them, okay, unless it's voluntary. Fake pills. That's right. Okay, fake pills or like, you know, sham treatment, right? Sham pills. These are maybe sugar pills or pills that has no known effect on the risk of catching an infection. And the purpose of having a placebo group is to account for bias and psychological effects. Okay, so there are your three groups. Right? So if we're doing a vaccine study, right? Control group would be no vaccine, treated group would be vaccine, and the placebo group would be receiving a vaccine minus the active ingredient. So if we were talking about the mRNA vaccine, it will be missing the mRNA in it. Okay, so those are the three groups. Part D, what is the Part D question asking? What factors must be kept constant between the various groups in your experiment? Okay, so these factors, and pay attention here, okay, because there is a question in the lab that involves this concept, and I want you to understand what I'm talking about. So these factors that must be kept the same, we call them standardized variables. Okay, so these are things that could change, but we want to keep them the same. Okay, so what's the point of having standardized variables? Okay, so between the three groups here, the control, the treated, and the placebo groups, we want to standardize everything else as much as possible, such that if there is a difference in the number of co-infections, we know that it was due to the supplements and nothing else. Okay, so that's why we need to have the standardized variables, okay, to make sure any observable difference in the dependent variable is actually caused by changes in the independent variable. Okay, so what I'm going to ask you to do is, give me a second. What I'm going to ask you to do is go to the chat box. I just posted a link, and I want you to go there, and it will ask you to list 3 standardized variable for this particular experiment. Okay, so type in 3 answers. It's completely anonymous, and then we will look at it as a class afterwards. Okay, so again, there is a link on the in the chat box. If you have a phone and you want to use QR code, here it is as well. I just need to share the screen. There we go. Okay, so I want you to click on the link and answer the question. And the question is, between the control group, treated, and placebo group, what standardized variables do we need to consider? Does everybody understand what I'm asking you to do? If you need clarification, please let me know. Not clear. Okay, so there is a link in the chat box. You just click on the link. Okay, just click on the link and it will take you to this page. And then it asks you for 3 standardized variable for the experiment we're talking about. So what you need to keep the same for those 3 groups, right? That's the question. So you're gonna type the answer here. Okay, just keep it short and simple and then press submit. Okay. I have about 6 people responding right now. Just gonna wait a little bit. Okay, couple, maybe a minute or 2 more, just to see if other people want to respond, and you should. Okay, so I know whether you understand or not. Okay, so here it is. That's what the 11 people who have participated, thank you, have said. Okay, so again, right, what is the question? Remember, we're trying to see if the multivitamin is going to help reduce the risk of catching a cold. So between the three groups, what do we need to keep constant? Okay, so the way that these things work is the bigger the word, it means many people mention that thing. Okay, so temperature, right? It's a little bit vague, but I guess I kind of get what you guys mean. Like you want them to be in the same kind of type of climate, right? You don't want to be doing the study, you know, you don't want to have the control group in Canada, and then you have the experimental group in, I don't know, like Mexico or something, okay? Because the temperature is very different in these two places, okay? You want them to be in the same geographic area. I suppose that's what you mean. And that makes sense, okay? And what else needs to be consistent? Diet is a good one. You want these people to be having similar diets, so we know the difference is not caused by, you know, different food that they eat. You know, we have environment that is good. Other medications, that's a good one as well. We want to make sure that none of them are taking any other medications or other supplements to keep it consistent. And here are common ones that people write down: pills and pills intakes. And then I saw one like time of the day taking the pills. That actually is not a standardized variable, okay? For it to be a standardized variable, it has to be something that is applicable across all three groups. Remember, only the placebo and the experimental group are taking pills, right? The control group is not taking anything, okay? So you cannot control the time of the pill taken if they're not taking any pills, right? Okay, so for it to be a standardized variable, it must be applicable to all the groups. Okay, so daily activities, that's a good one, right? You want them to be about the same body weight, right? Maybe similar representation of ethnicity, equal representation of gender, for example. Those would be example of standardized variable. Does that make sense? Does anybody have any follow-up question? There was one that says expose them to the same virus. Yeah, exposed by the same virus. We can't really do that, right? Like we cannot purposely expose them to the COVID virus and see what happens, right? Like think about how crazy it would be, right? To test the COVID vaccine, we give them a shot and then we purposely expose them to COVID, right? Remember, some people could be in the placebo group, right? You can't deliberately expose them to the virus, right? That is unethical. So that wouldn't be a standardized variable. Okay. Well, thank you for participating. We will now move on. Okay, we have a second question right here. A farmer wants to determine if feeding cows with high protein diet would increase milk yield. So the hypothesis, right? Oh, sorry. I don't think I finished this question yet. I went too quickly. What results would support your hypothesis? Well, if my hypothesis is that taking the vitamins will reduce the risk of catching a cold, then I would expect to see less infections for the treated group, right? And then the control group and the placebo group, I would expect to see more infections. And what will refute or reject the hypothesis? If the opposite is true, okay? If there are no difference between each of the groups, maybe 60% of everybody got sick in each of the group, or if people who take supplements actually got more cases of cold, then I know my hypothesis is not correct and therefore must be rejected. All right. Let's go to the cow one. A farmer wants to determine if feeding cows with high protein diet would increase milk yield. Write a hypothesis for this. Okay, so again, you either say high protein diet will increase milk yield or it will not. Okay, you pick one, you stick with it. Describe how the experiment should be set up to test this hypothesis. Okay, so again, I will have two groups. Okay, group one, we are going to, let's say we're going to call them the control group. Okay, control group. Okay, so what are we gonna give these cows in the control group? Someone please tell me. And then we have the treated group.
Protein.
We will give the control group proteins. Perhaps, and then Jada say nothing. We'll give them nothing.
Low protein.
No protein. Okay. We will give them normal diet. Let's say. Okay, normal diet. Okay, there might be a little bit of protein, whatever the normal diet is. Okay, and then for the treated group, it would be high protein diet. Right? So it will be everything else but extra protein. Okay, high protein diet. Okay. And then we will check the milk yield later. Okay, we'll find out if they need to, if they're going to produce more milk or not. Is a placebo needed for this experiment? Do we need to have a third group? Do we have to have some placebo, some cows in placebo group? Why or why not? No placebo. Okay, why do we not need placebo? Because they're cows. That's right. Because they're cows. Okay, no humans. You only need placebos for humans, right, to account for bias and psychological effects, right? The animals don't care if you give them a high protein or low protein. They're not going to be like, I'm going to hold out on my milk, okay, just to throw off your experiment, right? So for that reason, we do not need the placebo. No placebo. Okay, because cows don't have bias. Okay, they don't know. They're just animals. Very good. Okay, next one. Describe, actually, you know what? Before that one, we should do another poll. Here is another poll. Okay, go ahead, give that a try. Okay, of those people who've answered, I think most of you got it right. In this case, we have 1 ml of a drug diluted in saline solution, and then you're going to be injecting it into 20 pregnant mice. Okay, so what's a suitable control? Well, if we are using pregnant mice, then we cannot use male as a control, and we cannot use non-pregnant mice as a control. Okay. So control means no treatment, right? So we will not be giving them 2 ml of the drug. We'll just be giving them 1 ml of saline. That would be the correct answer. Okay. Moving on. Describe the main principles of experimental design. Okay, of experimental design. So couple things that we could talk about. For example, we want to have large sample size. Large sample size. Okay. Think of our vitamin experiment. We cannot just have 10 people in each group. That is too small of a sample size to make any kind of a definitive conclusion. Okay. If you think about the whole vaccine thing, when they were testing it in the clinical trial, right? You hear they say, oh, the Pfizer vaccine is like 99% or effective, or something very high, right? But then when it comes to the real world, then it turns out to be just maybe like 80 something percent, okay? And that is an example of the effects of sample size. Okay, even though in the trial they have thousands of people, which is a relatively big sample size, but in the real world, when millions of people get it, right, then the results are quite different. It's more accurate because more people are involved. So in any good experiment, okay, good experimental design, we want to use large sample size wherever possible. Okay, we want to have the proper groups. Okay, so include a control, having the experimental group, and also include a placebo wherever it's needed. We should also do it as a double-blind experiment, okay, where the participant does not know what they're receiving, and the researchers also not know what they're administering. Okay, some third party that has no stake in the experiment, they would hold the key to knowing who's receiving what. Another one is you want to make sure it is randomized group, okay, that you are not targeting a particular a group for your experiment. Okay, you want to have a good mix, good representation in your subjects, right, so that the results are as accurate as possible. So those are some of the things that we should consider when we designing an experiment. Next one, importance of including a placebo. We talked about that already, to minimize bias and psychological effect. And next one, what is the difference between blind experiment and double-blind experiment? So in a blind experiment, only the subjects, the people who are in the experiments, only they have no clue what they're receiving. Okay, I'm gonna give you a pill, okay, but you don't know whether it's a placebo or if it's an actual pill, okay? But the researchers, they know. In the double-blind experiment, nobody knows, okay? Again, the key is held by some third party, okay? So the experimenter, the researchers, they don't know what they're giving, and the people who are receiving it, they don't know what they're receiving. Everybody, it's like in the dark, okay, until the very end. And double-blind is the gold standard, okay? You want to blind the researcher as well because you want to minimize bias in the researcher as well, right? Sometimes there are like financial incentives and conflict of interest, that kind of stuff that could cause bias in the researchers. Any question before we move on to the applications? We're almost done. I know you guys have like an orientation at 2:30 or something. Alex, can you explain that again? Which part? The blind and the double blind? Is that what you? Yeah. Okay. So the blind means the participant, the people who are in the experiment, not the people who are conducting the experiment, but the people who are given the vaccine. For example, let's say we try to test the vaccine. Okay, so a blind experiment is when you are receiving the vaccine, you don't know if you're receiving the real vaccine or just a fake vaccine. Okay, that's a blind
question. Would like say the doctor or the pharmacist, whoever administering it, would they know if it's placebo or not?
In a blind experiment, they would know.
Okay.
The researchers, okay?
Okay.
In the double-blind experiment, they would not know. Okay.
Okay, perfect.
Yeah. Does that answer your question, Alex? Or was that Alex who was speaking? Sorry, I didn't pay attention to who was who. Okay, great. So again, right, we don't really do blind experiment. Okay, nowadays, like all the big pharma, all the pharmaceutical company has to be double blind. Okay, yeah, moving on. Application questions. Okay, so let's say I show up at your door. I'm a salesperson. I show up at your door, and then for some reason you open the door, and then I'm like, hey, I got this amazing lozenges, and it's going to help you cut down the number of days you will experience cold symptoms. The next time you have a cold, just pop one of these things and you will feel much better very quickly. And here is the data to back up what I'm saying. Okay, so I show you this graph, right? There's a nice placebo lozenge that I give to people, and you know, it takes them 7 days or 8 days to get rid of the symptoms. But if you take the lozenges, it will only shorten it to like 4 days. Okay, so what kind of additional information will you ask me, would you want, in order to improve the credibility of the results? What additional information would you like to see? Obviously, you should never buy anything from anyone that shows up at your door. But for the sake of discussion, what are you going to ask me about this graph? Nothing. Everybody just going to trust me? Slam the door in my face? Go away. Not going to buy it.
Sir, it's COVID.
Is that's right. Let's say this is pre-pandemic. Okay, what are you gonna ask me? There must be something you need to get, right? All jokes aside, this could be a multiple choice question, right? Which of the following additional information would increase the credibility of the study? Articles. Okay, Alex say articles. Yeah, maybe I pull out an article to show you. All right. I'll help you guys out here. Okay. For example, we talked about sample size. Right? Sample size. How many people were in this study? Okay, maybe it's just your family. Okay, it's 4 people, 5 people. Right? That's not credible. I want to know the sample size. Okay. Another thing is, yeah, age group. Okay. Was this done in otherwise healthy young individuals, or was it done in older people? What is the age group? Okay, I want to know other things such as who funded the research, right? Is there any kind of a conflict of interest here? Did the company who make the drug funded the research? Okay, was it a double-blind experiment? Okay. Number of days until cold symptoms are gone. How are they assessed? How are the symptoms assessed? Okay. Was it a self-reporting system? Did you have a doctor? Right? Yeah. Some people say the environment, same place. Were these people have the same kind of environmental exposure? You know, what's going on? So there's a lot of questions that you could ask to increase the credibility. Okay, they always have these graphs on those like those infomercials, right? Used to be more common, like you know, maybe 10 years ago, that they would have these infomercials that shows you all these amazing products with graphs and stuff like that. But a lot of them are just unsubstantiated claims. Okay, moving on. This is a very important one. I already explained this in the lecture recordings, but let's go through it one more time. Okay, there's a new drug called sleep, and it's supposed to help you reduce the symptoms of jet lag. Okay, jet lag is what you feel when you move to a different time zone. Okay, so if you travel to another place with another time zone, you might not be able to sleep at night because it's, you know, your internal clock is not synchronized, right? Okay, so, you know, I'm going to give you this pill, the sleeping pill, and I want to see if it's going to reduce the symptoms of jet lag. Okay, so here we have the control group. These are people who are not taking the pills. And as you can see, 70% of the people in this group experience jet lag, okay, symptoms of jet lag. In the treated group, right, only 40% of the people experience jet lag symptoms after taking the pill, okay? And then we have the placebo group, which is also 40%. So based on this graph, based on this result, would you say the drug is effective or not? Okay, let me quickly launch another poll. Oops. If you think it's effective, please choose A. Okay, the drug works, buy it. If you think it not works, choose B. And not sure.
So just to clarify, the control group are like, they have nothing. They're just
Nothing, yeah.
regular, right?
That's right. No pills were given to them. Okay, I'm gonna end the poll and share the results. And again, we are half-half. Okay? Half people think it works, half of the people think it does not work. So what's the answer? What if I ask you this on the quiz? What are you gonna choose? You are going to choose not work. Okay, and here is why. Let's, for a second, let's take away this bar for a second. Okay, let's just ignore it. Okay, let's not see this. Okay, no placebo. Okay, if you have the control group 70%, oops, and then the people take the pills, and only 40% of them say jet lag. Okay, so then it appears like it seems like it works, right? Like you give them the pill, they take it, and less people had jet lag, right? Okay, but we cannot just look at these two things. We have to also consider the placebo. What is the placebo? The placebo are the people who you give the pill to. You're like, take this pill, it will help you. So they take the pill, but you know it's a fake pill, right? Like the placebo group is receiving a fake pill. It's not the actual drug, right? So they take the pill, and then you also see 40%, only 40% of the people getting jet lag symptoms. So what does that tell you? It tells you it doesn't matter whether you give them the real pill or the fake pill. Both the fake pill and the real pill causes a reduction in jet lag, right? Which means your drug does not work. Okay, it's nothing special about the drug. I could have given them like a piece of candy, and then the jet lag would have come down anyways. Okay, so when the placebo group and the treatment group is functioning, having the same effect, right? Then we know it is not working. Okay, so here is the takeaway message. When placebo and treatment group both have similar results, okay, it does not work. Does not work. Okay, so in order for it to work, this bar would have to be similar to the control. Okay, so you give them a fake pill, you would expect it to not work, right? That's why the placebo group would have the same result as the control. Okay, so if this was the graph, then you can conclude that the pill works. Is that okay? All right. We have about 5 more minutes. Let's just quickly fill this in, and I will send you on your way. Okay, so first one, homeostasis is the ability to maintain a stable internal environment. This one goes from generalization to many specific observation. That is deduction. If you're going in the other direction, from specific to generalization, that is induction. A hypothesis is a tentative testable explanation, and it must be testable and falsifiable. Okay. The variable that is being measured or observed, that is the dependent variable. And then the one that is being changed or manipulated, that is the independent variable. We talk about a sham treatment or fake pill, that is a placebo. We need it to account for biases. If the subjects do not know if they're receiving a treatment or a placebo, that is a blind study. And then if the person who is conducting the experiment also not know, that is a double blind study. Relationship between two variables, that would be correlation. A primary source is a researcher's original report. Books, news reports, and magazines are examples of secondary sources. Okay. All right, that's it. Thank you very much for coming. I will see you again next Thursday, same time. And remember to watch the recording for lecture 2. I will post this recording afterwards and put the link on eCentennial. And I will also be posting the filled in version of the study guides. Okay, if you have questions during the week, feel free to contact me. Otherwise, have a fantastic day and have a good weekend.
Hi, sir.
Bye-bye.
Bye, sir.
Bye.